And then I asked my Dutch lab mates for help with pronunciation, but it's Huib, right?
Huib, yes, yes.
Yes.
Very
Okay.
Very good.
Kurt, we're going to test how you say free university in Dutch also.
Oh, they didn't let me, they didn't tell me how, they just said, don't bother to just say free university.
Yeah.
Yeah.
Smart choice.
Christian is back. You're muted though, Christian.
Yeah, just double checking. Everything's fine. Okay, you're live and things are running fine. Okay, I'll watch the YouTube and thank you so much, Hugh, for doing this.
My pleasure. Thank you.
Kurt, I had told Huib that I have to leave sharply at 1 o'clock. I have to actually start another Zoom meeting, 1 o'clock my time.
Okay.
So 10 o'clock your time. So I'm going to let you handle the questions, but just be cognizant of not letting this go on too long. You'll have to have the discipline to close it down, if not at 1 sharply, then at 1 or 2 minutes after the hour.
Yeah.
Okay.
We'll try to keep it on time for
Okay,
everyone.
very good. For those of you who've already joined us, we're still a couple minutes before the hour, so we're going to wait until about a minute after the hour and get started. So please stay on the line with us. Good day, everyone. It's just about the top of the hour. Let's just wait another 30 seconds or so before we get started officially. Okay, let's get started. Thank you everyone for joining us today. Welcome to Worldwide Neuro, a weekly series of lectures on the neurobiology of drug addiction. I'm going to quickly introduce Kurt Fraser, who just recently finished his graduate work in Patricia Janik's lab at Johns Hopkins University and is now a postdoc with Stefan Lamel at University of California, Berkeley. Kurt will then introduce our speaker today, and we're delighted to have Dr. Huib Mansvelder with us today. So Kurt, take it away.
Thanks, Eric. So it's my great pleasure to introduce our speaker today, Dr. Huib Mansvelder. Dr. Mansvelder completed his PhD at the Free University of Amsterdam in 1999. He then completed 2 postdocs, first one with Dan McGahey at the University of Chicago, where he studied drug-induced synaptic plasticity in dopamine neurons in the midbrain, in particular that evoked by nicotine. He then completed a second postdoc at Columbia with Rafael Yuste, where he trained in using 2-photon imaging to measure calcium dynamics in dendritic spines. He then returned to the Free University of Amsterdam in 2002 as a professor to start his own lab. He was promoted to full professor in 2008, and his lab has a particular interest in the interactions of prefrontal, cortical, and ventral tegmental area circuits, how they're interconnected, and how they're involved in behavior. His lab takes a multidisciplinary approach using a variety of techniques spanning electrophysiology and molecular manipulations, all the way to using these to better understand how networks of neurons interact during behavior. I'll keep it short and I'll let Dr. Mansvelder take over, and he's going to talk to us today, I think, about some exciting recent work from his lab looking at neural circuits underlying cognitive control.
Thanks very much, Kurt, for the kind introduction. And I also would like to thank the organizers for giving me the opportunity to present our work to you today. So, as was already introduced, in my lab here, we are interested in how the brain organizes cognitive control of behavior. And I thought I would introduce the topic a bit, getting already in the spirit of for this summer, I hope. We have a new Olympics again, if we get through this all right. But you have to imagine that cognitive, what I mean with cognitive control of behavior is that we control our goal-directed actions in such a way to allow them to happen in when the circumstances are appropriate, so that we can perform the behavior and reach the goals. As these swimmers, of course, are at the starting line and have to wait for the start signal to unleash their activity and win the gold medal. So from a very early age, we are all extensively trained in performing cognitive control of our behavior. So of course we have all kinds of goals. We want to ask questions, or we need to go to the bathroom, or we want to eat something. But we are all heavily trained in waiting till the circumstances are right, until we are allowed to show the behavior. And our society heavily relies on cognitive control of behavior. So as I mentioned already, to be a little bit more precise, so with cognitive control of behavior, I mean the organization of actions that are directed towards goals, so goal-directed behavior, and as well as inhibiting actions that are inappropriate. So this, in the context of drug addiction, failure of inhibitory control, failure of cognitive control leads to all kinds of problems. Eating too much, binge eating, obesity, excessive food intake, but also there's a high interaction between lack of or reduced cognitive control, impulsivity, and reduced attention and drug addiction, gambling, affective disorders, and other disorders, and neurological and psychiatric. So when drug users are asked to score their own impulsivity levels using the Barrett impulsivity scale, then what you will see, what is observed is that drug users report higher levels of impulsivity, so reduced ability to control their behavior when compared to their siblings and when compared to controls. And also these increased levels of impulsivity or reduced cognitive control is present in different subdomains of impulsivity. So subdomains related to attention and motor impulsivity and non-planning of behavior, so spontaneous actions. So drug users self-report higher levels of impulsivity, but also in laboratory animals, in rodents, impulsive animals self-administer more drugs. For instance, in cocaine self-administration, as observed by Jeff Daly, but also nicotine self-administration and also sucrose self-administration. Impulsive animals tend to self-administer more when they have the chance. So there's an interaction between impulsivity. There seems to be an interaction between impulsivity and drugs of abuse, but also drugs of abuse can induce higher levels of impulsivity. So in previous studies, we found that nicotine exposure at an early age during adolescence in rodents, in rats, leads to not only to reduced attention. Let me put this to laser pointed, to reduced levels of attention when measured in a 5-choice test, but also an increased levels of impulsivity in these animals when they were exposed during adolescence and tested in these behavioral tests 5 weeks after being 5 weeks abstinent. So apparently drug exposure can also induce reduced cognitive control. And when we further studied underlying molecular changes and underlying circuit changes, we found that in the medial prefrontal cortex, with proteomic analysis, that protein levels of the mGluR2 receptors were reduced when animals were exposed during adolescence, and they were reduced 5 weeks following exposure, and not when they were exposed during adulthood. And we could rescue attention parameters like accuracy in this 5-choice test. We could rescue attention parameters by applying this mGluR2 agonist to adolescent exposed animals. And when we in healthy animals applied in the medial prefrontal cortex mGluR2 antagonists, we had a similar reduction in attention parameters. However, these mGluR2 manipulations, these pharmacological mGluR2 manipulations in the medial prefrontal cortex did not rescue the impulsivity aspect of the changes that we observed, suggesting that these attention and impulse, that these sub aspects of cognitive control of behavior, that they are perhaps are regulated by either different neural circuits or different aspects of neural circuits. So that got us interested in how different circuits are involved. And of course, in addiction, but also in impulsivity, there's a whole range of brain areas, brain regions, and subregions of brain areas that are involved in organizing impulsivity, different forms of impulsivity. So waiting impulsivity, wait with the behavioral response until the circumstances are appropriate, or stopping impulsivity, so stopping an ongoing behavior. There are different brain areas involved within both of these impulsive forms of impulsive behavior. The prefrontal cortex features And of course, all kinds of connections from the prefrontal cortex to other brain areas and from other brain areas to the prefrontal cortex are involved in organizing cognitive control of behavior. So many of these, so this was reviewed in a very nice review by Jeff Daly, and but many of these identification of these brain areas that are involved rely on either lesion studies or pharmacological inhibition of brain areas. But when we look closely at, for instance, the prefrontal cortex, what we see is that, of course, when you silence, for instance, with muscimol, the anterior cingulate or the prelimbic area, you silence all the cells that are located in the different layers of these areas. And these pyramidal neurons, they project to different brain areas. So when you silence, for instance, anterior cingulate, you silence not only a projection to the striatum, but you also silence projections to the medial dorsal thalamus or the basolateral amygdala at the same time, when you just completely silence this part of the circuitry. So that got us thinking, perhaps what we need to do is look more in a projection-specific manner to the organization of cognitive control. And can we target neurons, pyramidal neurons in the prefrontal cortex that specifically project to another brain area, for instance, the striatum or the thalamus? So that's what I want to talk to you about today, is the studies that we've done recently, completed recently, to look at projection-specific role of pyramidal neurons in the medial prefrontal cortex, and look at pyramidal neurons that project to subregions of the medial dorsal thalamus, the lateral and medial part, and subregions of the striatum, the dorsal and ventral striatum. Also because, of course, the thalamus, of course, is heavily involved in organizing cognitive control of behavior, in the sense that a lot of sensory input go through that, and a lot of sensory input reaches the prefrontal cortex through the thalamus. And of course, the striatum is a very important, has a very important role in controlling behavioral output. So that's why we chose these areas. So Bastiaan Bruinsma, a PhD student in the lab, took up the first part of the study, and he first wanted to characterize the anatomical connection between the medial dorsal thalamus, subregions of the medial dorsal thalamus, and striatum. So he started out by just injecting retrobeats into different portions of the medial dorsal thalamus, so the lateral part. And in this case, he counted, he characterized the cell densities in the medial prefrontal cortex that were labeled by the retrobeats. So, and what he observed is that that in particular dorsal parts of the medial prefrontal cortex had neurons in them that were labeled by the retrobeats. You can see the quantifications over the different areas, and in all cases, it's different levels of the medial prefrontal cortex, and in all cases, it were the cell densities were highest in the dorsal parts, anterior cingulate, premotor area, in the medial prefrontal cortex. And also, he found that cell densities that were labeled were highest in the deeper layers of the dorsal medial prefrontal cortex. When he injected in the medial part of the medial dorsal thalamus, then he observed a different pattern, and now cell bodies were densities were highest at the ventral part of the medial dorsal of the medial prefrontal cortex, and but still they were located mainly in deeper layers, similar to the thalamic projections to the lateral part, and this was also similar to what was observed by Gabbett et al. Then he injected the dorsal medial striatum and found that again dorsal portions of the prefrontal cortex, medial prefrontal cortex, had cell bodies that were labeled, and the layer distribution was a little bit more even, but tended to more superficial layers. And for the ventral striatum, injections of retrobeats he found that the cell body densities were highest in ventral medial prefrontal cortex, which was also reported already, of course, and the densities were tended to more superficial locations. But all in all, so there seems to be also with the thalamus a gradient of projection neurons that target specific subregions of the striatum and the thalamus. Of course, it is in particular in the dorsal part of the prefrontal cortex. You see that we observe that the neurons that project to the thalamus, to the lateral part of the MDT, and the neurons that project to the dorsal part of the striatum are basically covering similar locations. So they should be intermingled, or potentially they could be the same pyramidal neurons. So the next question is, do individual prefrontal cortex pyramidal neurons project to both medial dorsal thalamus and striatum, or are these projections segregated? So to do that, Bastian took a combinatorial viral approach. So he injected mCherry and an EYFP containing vector that relied on different recombinases. So one is the Cre recombinase and the other was the Flip recombinase, and injected those Cre recombinase in a CAF2 virus in the lateral portion of the MDT, and the AAV retro Flip recombinase in the medial dorsal, or in the medial part of the dorsal striatum, and then looked in the medial prefrontal cortex whether mCherry and EYFP were co-localized in single cells or not. They also did a GAT staining on top of that to see if there was by chance any projecting GABA neurons, but didn't find any. But also all the EYFP and mCherry were basically segregated. There was only a very small percentage of pyramidal cells that were positive for both mCherry and EYFP, suggesting that that these projections to the striatum and the MDT were segregated in different pyramidal neurons. And similarly, he took the similar approach for the ventral medial prefrontal cortex, so injecting Cre recombinase and Flip recombinase either in the medial part of the MDT and the ventral striatum, and found that also here pyramidal neurons were either green or red, and did not show the blue GAT staining, and only a very small percentage of the pyramidal neurons had expressed EYFP and mCherry. So that led us to this schematic of connectivity between the medial prefrontal cortex and the MDT. So the dorsal medial prefrontal cortex tends to project to the lateral part of the MDT. So there's a gradient going to the ventral medial prefrontal cortex that tends to project to the medial part of the MDT. And for the striatum, this is of course more common knowledge, but it's a similar gradient. But the neurons tend to be more superficially located in the medial prefrontal cortex. The dorsal part projects to the dorsal part of the striatum, and the ventral part of the medial prefrontal cortex predominantly projects to the ventral part of the striatum. So the postsynaptic neurons that receive these projections in the thalamus and in the striatum, do these neurons have distinct properties, or are these synaptic inputs processed in similar ways in the different parts, subregions of these receiving structures? So Huib, another PhD student in the lab, took on this challenge, and so what he did is he labeled these projections with Chronos, which is a fast-acting general Dobson variant, and together with retrobeats, injected retrobeats in the medial dorsal thalamus, either the medial part or the lateral part, and he expressed Chronos in the medial prefrontal cortex, and then made brain slices of either the medial or the lateral of the medial dorsal thalamus, and recorded neurons that had the retro beats, so that were projecting from the thalamus to the medial prefrontal cortex, and checked whether they received inputs from the medial prefrontal cortex by shining blue light in the slice recordings, so in whole cell recordings from these neurons. We also filled the cells with biosighting to characterize their morphology. So morphologically, these neurons were the same. So thalamic neurons in either the medial part or the lateral part of the medial dorsal thalamus were morphologically very similar. Electrophysiologically, they also had synaptically, they were very similar. So synaptic inputs from the prefrontal cortex to these neurons had similar properties, but there were some parameters that were different. So neurons in the medial part of the medial dorsal thalamus had tended to have a higher input resistance and a slower charging kinetics, so a larger tau, time constant. And also looking at the firing properties, thalamic neurons in the medial part showed more action potential firing during steady state depolarizations, not during the burst, than the neurons in the lateral part. Taking a similar approach in the striatal neurons, not with the retro beats, because these neurons don't project back to the prefrontal cortex, but recording from medial spiny neurons and checking whether they receive synaptic input from the medial prefrontal cortex. Huib compared again the morphological properties between dorsal striatum and ventral striatum neurons were very similar, but electrophysiological properties of the neurons, the medium spiny neurons, were also very similar in dorsal and ventral. But now the processing of the synaptic inputs from the medial prefrontal cortex differed between dorsal and ventral medial spiny neurons, in that the ventral neurons showed the synapses showed strong or showed depression, synaptic depression upon repeated activation by chronos, whereas the dorsal medium spiny neurons, the dorsal striatal medium spiny neurons, did not show synaptic depression. So both of these results suggest that synaptic inputs and activity, initiation of activity in the different portions of the striatum and in the medial dorsal thalamus depends on the subregion. So inputs from the medial prefrontal cortex will be processed. My prediction is that inputs from these different areas will be processed in a different manner by these target regions. So what do these different projection neurons from the medial prefrontal cortex have in cognitive control of behavior? So to be able to test that, Bastiaan developed first an extended version of the 5-choice serial reaction time test. So when originally or classical forms of this test, they require extensive training of animals. And usually what's done is that an animal is trained by doing 1-hour sessions on successive days. And then it takes about 3 to 4 weeks when you use rats for the animals to reach a stable baseline at baseline parameters of this task. And that's a very long amount of training. And especially when you want to test successive different projection neurons in different groups of animals, that can take a lot of time. So inspired by our colleagues here in Amsterdam, who developed a homecage-based test for mice of this 5-choice test, we decided to take on this extension and develop it for rats as well. So Bastiaan developed an automated self-paced homecage-based test, 5-choice serial reaction time test. And basically what the gist of it is that the 5-choice chamber is connected to the home cage via a tunnel. So the animal can access the test whenever it is ready for it, whenever it wants. And basically the animal earns all its food by doing the test. So we did not food restrict the animals. they earned all their food by doing the test. And for those who do not know what the 5-choice serial reaction time task is, basically when an animal starts the trial by sticking its nose in the food magazines, that starts the trial, then there's a delay period, can be, it depends on the test, but between 5 and 12 seconds. I'll come back to that. Then in one of the 5 holes on the other side of the chamber, a light will come on for a short amount of time, and then the animal has some time to respond, to stick its nose in the lit hole, and then, if it does so correctly, receive a food reward at the other end of the cage. So it can make different kinds of responses, the animal. So it can make a correct response, as I just described. This is the rewarded response. It can make an incorrect response, which means it saw the light, but it's stuck its nose in the wrong hole. Or it can make an omission, which means that the light came on, but the animal made no response. So these parameters are classically related to attention aspect of cognitive control of behavior. The animal can also make an early response. So if they're very eager to earn the food reward, then they can make a premature response, which is related to impulsivity, so lack of cognitive control of behavior. So make a response before it's appropriate. And that's usually punished by a timeout. So classically, as I mentioned already, classically, in the classic version of the conventional training, it takes animals about 3 weeks on average to reach stable baseline. If animals have, in our hands, have unlimited access to the task, they learn it in 3 days. They can do it the full test and earn all their food through the 5-choice. But what we observed is that when they have 24/7 access, animals start to tend to make a lot of omitted trials. So as if they are not so, how I say, they're not, they can always come back later. So they start a trial and the trial runs, but they are not interested and come back later for the next trial. So then we started to time restrict the activity of the task. So for 2.5 hours at the start of the dark phase, animals were allowed to do the task. I mean, they could access the chamber always, but only in the first 2.5 hours of the dark phase, animals would be able to earn food by doing the task. And then we found that the omissions went down drastically, so back to conventional levels. And animals learned to do the task within about a week, so 7 days or so, which is still a strong reduction of the training time. And also for each of these 2.5-hour sessions, animals completed many more trials than in the conventional training. So conventional training usually is 1 hour in the task, and then 100 trials, complete 100 trials. But animals that had the time restricted access, they completed 400 to 500 trials in each session. So that also gives us a lot of more statistical power to do different tests. Okay, so using this task, we started to manipulate the different projections from the medial prefrontal cortex to either the thalamus or the striatum. And so we expressed inhibitory DREADD receptors in these projections by using a similar CAF2 approach, injecting CAF2-CRE in the target area, lateral portion of the medial dorsal thalamus, the medial dorsal thalamus, or the medial portion, the dorsal striatum and ventral striatum, and injecting the CRE-dependent DREADD receptor factor in the different parts of the medial prefrontal cortex. And this needle is on the wrong side; it should be here. So we did it on both sides. So, and then after successful expression, after some time, we took the animals and put them trained them in the self-paced 5-choice task. And as you can see here, so this in this example, an animal had completed 450 trials. Oh yeah, I should also say that in order to really test whether they were cognitively controlling their behavior, we made the delay period, so the period that they have to wait before the light comes on, unpredictable for the animal. So we had three versions: a 5-second delay, 7.5-second delay, and a 12.5-second delay, and they were randomly varied. And here you see an example of the different responses the animal made. So it made correct responses. So this lasts 2.5 hours. Correct responses, incorrect responses, premature responses, and omissions. So you see that for all the different response types, they're relatively equally distributed across the 2.5-hour session. And of course, in these, what's also reported in the conventional 5-choice, what you see is that when you make the delay period unpredictable, then animals start to make more premature responses, in particular on the longer delay times, where they have apparently a more difficult time inhibiting the urge to respond. So then we injected CNO just before these 2.5 hour, just before the dark phase, and at different concentrations, so either 5 milligram per kilogram or 10. And we also had an EYFP control group, and we expressed the results as a change in percent of, for instance, premature responses relative to 0 milligram per kilogram. So so what's the change in responses induced by CNO injection? And then what we observed is that for the different projections, so this is the medial dorsal thalamus with the lateral part, and this is the medial dorsal thalamus medial part in green. What we observe is that the medial prefrontal cortical projections to the lateral part, when you inhibit those with these DREADD receptors, then the amount of premature responses go down. And for the projections of the medial prefrontal cortex to the medial part of the MDT, the premature responses go up. So, and actually for the attention parameters, we only saw a change in the percentage of omissions, so more omissions when we inhibited the medial prefrontal cortex to lateral part of the medial dorsal thalamus neurons. Then we saw an increase in omissions, but in the other groups we did not see a change in the omissions. So this suggests that during this preparatory attention phase, when the animals are inhibiting their urge to respond, that this dorsal medial prefrontal cortical projections to the lateral part of the medial dorsal thalamus take care of a sort of a go signal. So and that the ventral part and to the medial part of the MDT take care of a stop signal. So wait until the time is right, until the cue is there. And then similarly for the medial prefrontal cortex to dorsal striatum and ventral striatum, we injected in similar ways and found that only the projections to the dorsal striatum from the dorsal medial prefrontal cortex, when we inhibit those, that we see an increase in the premature responses, and none of the projection neurons to the striatum, when they were inhibited, changed the attention parameters. So emission, in this case omissions, but the other attention parameters were also not affected, suggesting that also the dorsal the projections from the dorsal medial prefrontal cortex to the dorsal striatum signal a stop for the behavior, for inhibiting the behavior during the preparatory attention phase. So that gives rise to, let's say, a bidirectional circuitry that is anatomically segregated. So between the dorsal and ventral prefrontal cortex, but also between the deep and superficial layers, where the projections to the thalamus in the dorsal part signify go, whereas the dorsal neurons, the pyramidal neurons that are more in superficial layers that project to the dorsal striatum, signal a stop. And then with the ventral part, the thalamic neurons, there's also a, let's say, bidirectional regulation that is between the projections to the medial and lateral parts of the MDT. So then the next question is, of course, I mean, it's all very fine. There's this inhibition, inhibitory manipulation, and then changing the behavior. But can we actually also see part of this back in the activity of the neurons? So when are medial prefrontal cortex pyramidal projection neurons active during cognitive control of behavior? Are they actually recruited during this behavior? So to test that, we took two approaches. So either we used fiber photometry of GCAM, or we recorded wirelessly with silicon probes activity of optotagged projection neurons. And you see an example of that here. So I'll start first with the optotagged neurons. So we expressed, or Huib Terra expressed channelrhodopsin in projection neurons to the dorsal part of the striatum. And when he turned on the laser, he can record induction of activity in some of the neurons, not all neurons, but some of the neurons. And that's depicted here as well. And what he observed is that of these optotagged neurons, these projection neurons, that there are three kinds of responses. So some of the neurons, they are persistently activated during the task. So when the animal initiates the trial by sticking its nose in the food magazine, then there's a moment that the animal turns around and starts to observe the 5 holes. That's what Huib calls cue orientation. So for all the animals, he characterized for each trial when the animal turned around, and that's the green point here in the histogram, and in the event plot. So each of these little dots is an action potential, is a spike, and these are the different trials plotted on top of each other. This is the moment of the cue orientation, and this is the moment of the response. And in this case, he only plotted responses, trials with correct responses. So when you align according to the cue orientation, then you see this sharp increase of activity of these neurons at the cue orientation moment, and a sharp decrease of the activity at the response moment. And during the delay period, these neurons tend to be activated. Then there's a group of neurons that was not activated during the delay phase, and then there was a group of neurons that was silenced or reduced activity during the delay phase. So this delay-related activity also changed when we made the delay period unpredictable to the animal. So the animal had to inhibit its behavior for an unpredictable amount of time, either 5 seconds, 7.5 seconds, or 12.5 seconds. And we saw that the activity of the neurons just continued until the response moment, until the behavior could be unleashed, basically. And similarly for the inhibition. So the delay period, the length of the delay period also determined, or was correlated with the length of the inhibition of activity. And this is for all the 44 opto-identified neurons that Huib recorded. And you see that the delay period is similar to the activity and silencing period. And actually, of course, Huib also recorded many neurons that were not optogenetically activatable. So these were non-identified neurons, and they also show similar responses, so either a decrease or an increase in activity. And what you see here is the 3 different behavioral points. So start of trial, cue orientation, and response is 1, 2, 3. And red means activated, and blue means reduced firing rate. But what we observe is that in the projection neurons, the proportion of activated and silenced neurons is larger than in the unidentified population. So this is quantified here. So the fraction of neurons per group. You see that in the non-identified neurons, there's a lower percentage of activated, a higher percentage of non-activated, and a lower percentage of inhibited, silenced neurons compared to the opto-identified frontostriatal neurons. And what about, can you then tell something about the trial outcome by looking at these at the activity profiles of these of the opto-identified neurons, of the projection neurons? And what Huib found in short is that persistently activated neurons showed reduced activity during premature trials. Persistently silent neurons showed less silencing during premature trials compared to correct trials. And this is quantified here. So they show a reduced level of activity and level of silencing. And also the neurons tend to start firing or start being silenced earlier during premature trials. And that you can see here. So this is again the moment that the animal turns around and starts to face the cue holes. And on premature trials, animals turn more quickly earlier than on correct trials. This is in the behavior, you can already observe that. But also in the firing rate, what we see is that the increase in firing rate during premature trials increases before or earlier than during correct trials. And the same holds true for the silencing, which I'm not showing here. So on average, the response types, when we distinguish premature trials from correct trials, it looks like this. So during premature, when an animal is going to make a premature trial, it turns around more quickly earlier. The neurons, the projection neurons to the dorsal striatum start to be changing their firing rate, either increasing or decreasing them earlier than during correct trials, and also to a reduced level. So the increase and the decrease are both reduced. So these neurons, the activity of these neurons tells something about the trial outcome that the animal is going to do behaviorally. So when are the other projection neurons active? Do they show a similar type of firing rate change? So this is the work that Siebren de Kloet did in the lab. So he labeled the projection neurons with all the 4 different projection neuron types with GCAMs in a similar way as I described again before. And this top portion you already saw, but here you see the fiber placement and the fluorescence in the neurons. And what you see below here is the GCAM signal during the behavior. So the animal is doing the 5 choice. You see that the calcium or the fluorescent levels go up and down. When he used an EYFP control, you see that the fluorescent level stays low during the behavior. Sorry, but with GCAM you see all these changes, and these were the placements of the fibers and the fluorescence locations in all the different animals. So this is what the activity profile looks for the different projections, and again we have the short delay trials, the medium delay trials, long delay trials. They're unpredictable to the animal, so it's variable ITI for the aficionados. And what we see is that basically for the medial prefrontal cortex projections to the thalamus, medial part, the lateral part and the medial part, and the dorsal striatum, basically the profile is roughly speaking similar, so they go up. Fluorescence, GCAM fluorescence goes up, but only for the ventral medial prefrontal cortex to ventral striatum, we did not see changes in activity, or changes in fluorescence, I should say, because it's an important difference. So we did not see changes in fluorescence during the delay period. But the increase in fluorescence we observed in the other projections followed again the length of the delay period, suggesting that these projections are changing activity depending on the length of the delay period, as long as the preparatory attention phase lasts. But what we did observe was that there were subtle differences in kinetics in how fast the increase occurred. For instance, compare here this blue trace versus this green trace, and the level of change was different between the projections. So Sebren quantified that in very many ways, comparing the different projections, but also comparing different trial outcomes, so correct trials, omissions, premature responses, and then for the different projections. And these are the fluorescence profiles averaged and plotted for the different response outcomes of the different projections. You see that there are differences between the projections. And with the permutation tests, he tried to statistically figure out where these traces are actually different. So he finds that there are at several between the particular the ventral striatum and the other areas there are pronounced differences, because ventral striatum projecting neurons did not show fluorescence changes during the delay period, but also with respect to the other projections between each other, there were differences at various points during the different trial outcomes. But of course, the important part is, so first of all, are there differences? So there are some differences. And the important part is, can you, from the activity in these projection neurons, can you then predict the behavior? So that was what Sebren did finally. So he compared first differences from baseline between the different trial outcomes, but also then the differences between the trial outcomes in the different projections. So, for instance, for the dorsal medial prefrontal cortex to the lateral part of the medial dorsal thalamus, can you see differences between the fluorescence traces when you compare correct trials outcomes and omissions, or correct trial outcomes and premature responses, or omissions and premature responses? And what he finds is that for this projection, the fluorescence trace is different, especially during when you compare correct trials with premature trials. And for the ventral medial prefrontal cortex to thalamus projection, there are long periods where the differences are between the correct trials and the omissions, and a couple of moments that the correct trials and premature responses differ. And then for the dorsal medial prefrontal cortex to striatum, in the fluorescence traces, he finds only statistically significant differences between the correct trials and the omissions. And he didn't pick up differences in the correct trials versus the premature responses as we did in the electrophysiology. The ventral medial prefrontal cortex to ventral striatum projections did not show any differences between the trial outcomes in fluorescence profiles during the delay period. So, and especially when you combine the information, activity information based on the fluorescence between the different projections, then definitely you can you can predict trial outcomes, differences in trial outcomes between correct and premature and correct and omissions based on these data. So that's basically what I wanted to show you. Of course, so to summarize what I told you, so we looked at projection-specific roles of medial prefrontal cortex neurons to the different portions of the thalamus and different portions of the striatum in organizing cognitive control of behavior. What we found is that the projections to subregions of thalamus and striatum are, first of all, anatomically segregated. I mean, so this is of course overstating a bit because they are intermingled, but the density of the neurons is more towards deeper layers for projections to the thalamus, more towards superficial layers for projections to the striatum. But neurons are definitely intermingled. And that's why it's, I think, useful to look at projection-specific properties. What we find is that in the target areas, both synaptic and cellular properties can differ between pathways. So between the dorsal and ventral striatum, we saw there was a difference in synaptic depression in synapses received from the medial prefrontal cortex. And in the thalamus, we saw that there's differences in cellular properties. So in the amount of spikes that are being generated when these neurons are depolarized, they're much more excitable in the medial part of the thalamus than in the lateral part, which will result definitely in different ways of processing of information from the medial prefrontal cortex. So we find that there are different, let's say, pairs of activities, of projections that allow bidirectional command of cognitive control of behavior. So between the dorsal and the ventral part, when you look at the thalamic projections for go and stop signals, and between the superficial and deep layers, between projections that go to the lateral part of the medial thalamus and the dorsal striatum, there also seems to be a bidirectional command of cognitive control behavior of impulsivity. So we found that projections are active during this preparatory attention phase, suppressing behavioral responses. They show different kinetics in activity, so some come a little bit earlier, are activated a bit earlier than others, and the activity in these projections predicts a behavioral trial outcome. This is most clear when you look at the electrophysiological data, so the wireless EVs recordings had a much better resolution, but also in the fluorescence we could find evidence for prediction of behavioral trial outcome. Of course, looking at only 4 projections from the medial prefrontal cortex is a huge simplification or a huge, let's say, underestimation of what the prefrontal cortex can do, because, of course, it's connected to so many more areas. And we know, of course, already that other projections to, for instance, the lateral hypothalamus and the subthalamic nucleus are also involved in impulsivity, as was shown in this paper from Yang Dan's lab. But there are, of course, many more projections from the prefrontal cortex to, for instance, amygdala, but also sensory areas. So, which other projections should we consider? Could we somehow consider them all? Would be a great question to address. Of course, I didn't touch upon how much overlap there would be. Can we, from these projection-specific studies, better figure out what the overlap and the differences are in the circuits that control different aspects of cognitive control of behavior, such as attention versus impulse control? That would be great to figure that out. We found that one projection to the lateral part also increases omissions when it's inhibited. So possibly having a role in attention behavior as well, not only impulse control. Of course, I didn't get to the part whether these projections in their activity are altered by drug exposure. This will be a great project to do. And are there sensitive periods when animals are exposed to drugs of abuse during adolescence? Is that different from adult exposure? And of course, do changes, if any, in these projection neurons, do they last or not? So these are of course, points that we would love to study, and I would love to tell you about it in the next talk. But for now, I would like to thank you for your attention. I would also like to thank the people that did the study, in particular Bastiaan, Huib, and Sibren, PhD students in the lab, but of course they received help from many other people in the lab and also outside the lab. So in our institute, Tommy Patti, in particular, Marcel van der Roest and Rogier Min and Maarten Loos. And I would also like to thank the people that fund our work, or the institutions that fund our work. And thank you for your attention, and I'm happy to answer questions.
Thank you so much, Huib. That's for that great talk. I'm going to turn things over to Kurt to moderate the question and answer period. I would like to remind people to put their questions in the Q&A function on Zoom. Kurt, take it away.
All right, great talk. I think I'll start with a question, which is thinking about these different outcomes in the trials and looking at the fiber photometry signal, and even in the electrophysiology study as well. I was wondering if you ever looked at activity based on like a trial history basis. So say if the previous trial was an omission, does that alter the circuit dynamics in certain projections? It's kind of curious to me too that the incumbents projecting PFC neurons are completely silent, it seems like, during the photometry recordings. If you could comment on what you think that's doing.
Well, these are multiple aspects, but indeed, the last point, we were also very surprised that the frontastriatal projections were, first of all, when we manipulated them or inhibited them, that they didn't show an effect on behavior, but also that we didn't see any calcium or fluorescence activity during the delay period. We have a couple of technical aspects that we should have compared core and shell projecting neurons. Perhaps we went wrong there, or we missed them, but that's also something to figure out later. I know that from talking to Over Yisar at Weizmann, that he also studied these ventral striatum projections from the medial prefrontal cortex and also had a hard time finding a functional role in the 5 choice. So perhaps there's something there. But we don't know. To relate to trial history, so indeed, so using this combi cage or this home cage based task, we get so many trials per session, and also we can do the throughput of the amount of animals is also much better. So we definitely have statistical power to compare trial history, and also in the DREADD experiments. So we are preparing a manuscript that looks at that, and then we find that when we inhibit, in particular, when we inhibit first of all, of course, in general, the behavior of the animals indeed improves after an error response, but also after a trial difficulty. So when an animal is going through a long delay trial before, and then the next trial it performs better, more correct responses. But also when we vary Q durations, so the light durations from very short to longer, also this difficulty improves behavior on the next trial. And then when we inhibit the different projections with the DREADDs, we find that only when we manipulate the dorsal prefrontal cortex, medial prefrontal cortex to the lateral part of the thalamus, that projection, when you inhibit that one, that affects or impairs the error, the self-monitoring and the trial history and the improvement of the performance in the next trial. Yeah, so there's these projections to the thalamus at least also play a role.
And a last question, I think that answers the question also from Hiro Morishita in the chat. So a last question is, do you think that these are recurrent, say, corticothalamic loops where the thalamus is projecting back into different areas of cortex, or even back into the same parts of prefrontal cortex? Or do you think that this is more of a distributed network-based system where, say, they converge downstream onto areas like the amygdala, potentially, that are engaging in sort of self-monitoring behavior?
Yeah, that's a great question. I would love to know the answer, but many of the neurons that we recorded from in the thalamus had projections back to the prefrontal cortex. So it was and received inputs from the prefrontal cortex. So I think that many of the thalamic neurons are part of the recurrent aspect of networks, but this is definitely something that we need to test, but it's a complicated question.
Okay, and I think we all have things to get to, so I think we're gonna stop there. So Eric, do you have any last words?
I just wanted to thank you again for a great talk, and thank you everyone for joining us today. Stay tuned for next week for the next lecture for this neurobiology of addiction series. Thank you all very much and have a great rest of the day.
Thank you.
Thank you all.